Hello, everyone, and welcome to Learning and Mending Post-Human Infrastructure. Tonight, we'll start off with welcome you all, and I'd like to share the embroiderer of the week. And this one was sent back all the way from New York City, and it is by Melinda Ring. And I thought it was perfect for tonight's reading. It is a jack-o'-lantern mushroom, and it's just so beautiful. When I look at the close-up, I don't know if you can see what I see. Okay. So many tiny stitches and this beautiful shading. And I think I was trying to dissect it earlier. It looks like she took some of the floss and braided it together and then sewed it in, which I thought was a really cool technique. So this is tonight's embroidery of the week. So we thank you, Melinda. And now, while we're all kind of gathering into our squares, I thought it'd be great for us to kind of do some popcorn introductions, say our name, where we're zooming in from, our pronouns, and what we are working on tonight. Some of us have embroidery kits, some of us are waiting for embroidery kits, and some of us are working on something completely different that has to do with mending. So please share whatever you have. I am working on my Mighty Buffalo is coming along one piece at a time, but it's getting there. I'm hoping to make this part this week. So my name's Jen, and I'm zooming in from Studio City, and I use she, her pronouns, and I am going to pass it over to Lee Adams.
Hello, I'm Lee Adams, and I use she, her pronouns, and I'm zooming in from Altadena, and I'm working on Misha's beautiful corn. Not quite doing it justice, but I really do love this pattern enormously. And I am going to zoom it over to Alexandra.
Sorry. Hi, I'm Alex. I'm calling in from Providence, Rhode Island. Today I just want to do some intuitive drawings based on the readings. I'm going to pass it on to Cindy.
Hi, I'm Cindy. I'm calling in from West Covina, and I will be working on a crow embroidery that from the lost learning and mending. And I will pass it along to Juanita.
Hello, my name is Juanita, and I'm calling in from Glendora, California. And I go by she, her, and I'm working on the corn. It's my first time actually mending, and I'm doing it so small because I think I'm too focused on the details, so it's taking me forever to finish. But I'm having a lot of fun with it. It's a new creative outlet, and yeah, it's giving me inspiration for other projects. So I will popcorn it. How do I see? Okay. How about I send it over to Millie Mason Moore?
Thanks, Juanita. Hi, everyone. I'm Millie. I am currently sitting at the Moon, one of our places that Metabolic Studio tends the land at for Undevelopment 1. And I will be sitting with the aromas of these beautiful marigolds. Yep, there you go. You can see them. They smell so good. We'll explain why some of us have marigolds in our background in a little bit, but I will now pass it on to Shelley.
I'm in La Crescenta, California, and right now I'm playing with my feral kittens. This one still needs a home. if anyone wants. She's lovely. The others are adopted. And I will pass it along to Kumar.
I think Kumar said in the chat that they can't speak.
Yeah.
They're in a library. But I'll go if that's okay. I'm James. Pronouns are he, they. I live zooming in from Los Angeles, Gabrielino Tongva Quiche territory. And tonight I will be reading about fungi with everybody. Very excited to be here. And I will pass it on to Lauren.
Good evening, everybody. I'm Lauren. I use she, they pronouns. I'm calling from Tongva Gabrielino sacred land on what would have been the floodplain of the Unbridled River. And I am going to be sifting through pine resin I collected at 10,000 feet last summer to take to an altar that I'm making tonight. At the Undevelopment 1 site, where we're marrying our ritual of honoring the people we've lost this year with the newly liberated floodplain that has been brought out from under the concrete. So yeah, if you want to join in, we posted a whole bunch of virtual backdrops in the chat bar if anybody wants to choose one and have a Dia de los Muertos backdrop. We can all be in the same virtual space. So I pass it on to Laura O'Connor.
Thank you. That's a gorgeous backdrop. And greetings on Dia de los Muertos. I'm down in Kumeyaay territory. Laura, I use she, her pronouns, and I'm very honored to be with you all on this auspicious day and in general. And I came on late, so Kavi, I'm going to hand it off to you. If you've gone, maybe you can pass it on.
Yeah. Thanks, Laura. Hi, everyone. I'm calling in from Sacramento, California, the Plains Miwok territory and the ancestral home of the Mitchell Wilton Rancheria. Use he, him pronouns, and I'm not doing any mending tonight, but I'm hoping to do just some learning. So I'll see you all in the chat and looking forward to tonight's reading. I'll pass it over to Ashley.
Hi, everybody. I'm Ashley, she, her pronouns, and I'm over in the city of Alhambra. And tonight I'm going to be working on mending an urban site for emergency homeless housing while I listen in. So that's me, and I'm going to pass it on to Stefano.
Hi, Ashley. Hi, everyone. I'm Stefano from El Sereno. Excited to this new episode of Learning and Mending with James. And as Lauren was saying, if you want to put an ofrenda background image on your Zoom, you can grab it from the chat room. They said, I'm going to pass it to Yeva, if he's there.
I'm here. Hi. Hi, I'm Yeva. I'm calling in from Glendale. I use she, her pronouns. And tonight I'm working on the corn, making some progress. We'll see how long it takes me to finish this. But I'm going to pass it on to Dee.
Hi, Derainer Holland calling in from Baldwin Hills, California, and I go by she, her pronouns, and I am working on this gorgeous corn piece by Michelle, and I have a lot more to do, but I'm happy to be here tonight, and I will pass it on to Alex.
Hi, everyone. I'm Alex, and I'm calling in from Mar Vista, Los Angeles. And I am too working on my corn, and it's going to take a while, but it's going to be really fun to get to the little corn parts. And looking forward to the reading with James with this episode. So I'm going to pass it on next to Doug Lee.
Thanks, Alex. My name is Douglas Lee. I'm calling in from the Metabolic Studio on Tongva Gabrielino land. I use he, they, she, why not? Anything. The universe is complex. Pronouns. And tonight I am just listening and learning and very stoked to hear James's curated readings. So with that, I will pass it on to Aaron Thompson.
Hi, I'm Aaron Tupag Thompson, and I'm calling in from Mar Vista, Tongva Kish, Chumash land. And I am weaving together a gathering tonight for the Day of the Dead over matsutake and a bunch of mushrooms. I am really excited that once again, y'all's reading group is overlapping also with my reading group, Exploring the Mycoverse. We're currently reading Entangled Life, and I just spoke at the Radical Mycology Convergence. So I'm really stoked James is speaking tonight. Happy to be here. And I am going to pass it off over to, did Andrea go yet?
Hello, I'm Andrea, she, her pronouns, and I'm calling in from Los Angeles. And I'm going to be working on some things for Maya Frenda tonight. And then I think I saw Jen wave her hand.
That is correct. Are you done? Sorry. Okay, I will take it. And I just want to welcome everyone again to our 9th edition of Learning and Mending Post-Human Infrastructure. This series will explore the commons of soil, the air we breathe, water, sound, and the potency of seed and mycelium networks. It involves letting go of the outdated ideas that do not support and serve the life web. As a studio practice, we aim to facilitate this rebound and regeneration. We seek to engender self-propagating and self-generating feedback loops that feed the life web in places that have been ground down to an inert state. Artists need to create on the same scale that society has the capacity to destroy. Posthumanism is a concept that critiques anthropocentrism and the philosophical branch of humanism, a dominant ethos that has shaped social relations and the way humans relate to the non-human world for centuries. There is a rich discourse on posthumanism and posthuman. Therefore, there are many ways one might define these concepts. Posthumanism pushes back against the centering of the human and moves towards thinking that eradicates hierarchy of being and challenges the life, non-life binary. Posthumanism is an ontological pursuit of what it means to be human, what it means to be non-human, and where these categories rupture and collide. This is our 7-week series and will be facilitated by Millie Mason Moore, Metabolic Studio Farm Lab Coordinator. We are inspired generalists rather than experts and hope to facilitate an open community dialogue. This series has run each Tuesday with the exception of skipping November 8th and will conclude on November 14th. If you'd like to receive an embroidery kit from us, please send your name and mailing address to info@metabolicstudio.org. Tonight, we will share our screen displaying this week's reading. Feel free to jump in and read aloud from the text as you feel inspired, as we welcome one and all to both listen and giving voice to the unique text we share each week. And now I will pass it to Millie.
We're actually going to pass it to Kavi. Kavi has a poem that he will be reading. Thanks, Jen.
Everyone, hope you all are comfortable and settling into wherever you are and ready to take in tonight's reading and participate in our discussion as well. I'll start by sharing a poem offered to us by our very own Izzy Santana, titled An Offering to Decay. I want to decay like a fallen tree, fungi growing in my crevices. An oyster mushroom replaces my tongue, my flesh taken up by lichen. I offer my blood to the soil as it holds my decaying body like a newborn baby. My cries become absorbed by the sound of thunder, tears shed alongside the rain. What is the difference between varicose veins and rhizomatic canals made by falling water, and patterns of sand formed by wind and Lichtenberg figures and neural pathways? I would like to discard my brain as sustenance for a passing vulture, or a scrumptious treat for a hungry coyote, a pile of mush for all scavengers to delight. Decomposers work magic as gut-wrenching smell transforms into emergent, nutty, rich, wet clay and sweet terrestrial matter. A series of brief moments, arrangements of possibilities, bloodshed and passing sighs, the only end to become once more.
Thank you, Kavi. Thank you, Kavi. Thank you, Izzy, for writing that beautiful poem. Great evening, everyone, and welcome back. I'm Millie, and I'm always thrilled to see everyone virtually, even through these little boxes. This series of post-human infrastructure was collectively curated by our team at Farm Lab. Last week, we read about compost, and our colleague Mish, who is also here, described the many uses Farm Lab undertakes with this rich and full of life medium. As mentioned, compost serves as one of the base materials we use to initiate many of our case studies here at Metabolic Studio. Tonight, I'd also like to bring attention to the marigold. Marigolds. Today is a big day for the marigold, as many of us celebrate Dia de los Muertos. It symbolizes the beauty, the fragility of life. Marigolds are believed to help guide lost souls. On Dia de los Muertos, they help guide our loved ones from the other side to the celebration with us, and then back again to their resting place after the party is over. Some say it's that signature pungent smell that helps guide the way. The marigold is also an important flower in Southern California and in the garden. They are native to Mexico, but have been cultivated around the world. Because they are hardy and drought tolerant, they do well in Southern California climate. Marigolds are also fantastic for the vegetable garden. They make great companion plants, deter some pests, and they're also edible. Plus, they just add the beautiful burst of color to any garden. And of course, marigolds are also compostable. In this week's post-human infrastructure, we will explore the fungal queendom in order to imagine the future that fungi will inherit and mold, a glimpse into the Earth's deep past and the earliest natural history helps envision a post-human world. Over the eons, we will see fungal dominance during extinction events. Their opportunistic nature is extreme in extreme environments. 40-foot fungal giants and how organisms that pair with fungi are shepherded through uncertain times. Here is a prompt and some guiding questions for our casual dialogue after the reading. Farm Lab investigates and works with the queendom of fungi on site through microscopy, inoculating logs, and culturing fungi. How do we interact with invisible webs and gardens like mycorrhizal networks? How do we observe, hold space, appreciate and recognize living systems that are always felt but not directly seen? How do you engage with decomposition metaphorically and literally? Do you have cycles of breaking down your practices, emotions, beliefs, experiences, and relationships? What is the fruiting body or bounty that follows in its wake? Now, I'd also like to mention, as I always note, we are inspired and passionate generalists rather than experts on these following subjects. Should you not feel comfortable having a conversation when the time comes, just feel free to use the chat bar to add your questions, thoughts, dreams, or resources. Also note, anything added to the chat bar will be included on our Discord Water Community Power. I believe Matt, should be here, will be now adding the Discord link to our chat bar. The Discord has been established to continue these dialogues and to open source our collective knowledge. Now, I would love to introduce our colleague, James Oliver. James is a resident and radical mycologist at Metabolic Studios Farm Lab. He's an advocate for fungi, some of the most neglected and poorly understood organisms. James's work explores the many roles that fungi play in remediation, restoration, food systems, and soil building. Let's welcome James with a virtually round of applause. Thank you, James, for being with us. And one question for you, James, that I ask all of our other team members before we begin reading. Can you briefly describe your experiences working with fungi at Metabolic Studio?
Sure, absolutely. Well, there are a lot to talk about, but I think one of the most poignant ones is exploring something we're going to be reading tonight, which is mycorrhizal fungi. And Lauren mentioned earlier, celebrating the Day of the Dead with the floodplain, which is at one of our sites. Is uncovered at one of the sites that we're working on. And, you know, the history of the LA River is one of industrialization. And so finding mycorrhizal fungi for the first few years that I was there before we had encountered the floodplain was very difficult. And mycorrhizae, they're not like typical mushrooms. They don't fly in the wind, so they really suffer from habitat fragmentation. So but they're really important for establishing networks underground that help plants resist disease and drought and things like that, and also sequester carbon. And I will say that my virtual background is an example of the mycorrhizae at the floodplain at our site. So that's just one of the things that we've done together.
Awesome. Thank you, James. All right, without any further ado, Emily, would you be so kind to share our reading tonight?
What is it like to be a fungus? There are moments in moist love when heaven is jealous of what we on earth can do. Hafiz. Fungi are everywhere, but they're easy to miss. They're inside you and around you. They sustain you and all that you depend on. As you read these words, fungi are changing the way that life happens, as they have done for more than 1 billion years. They're eating rock, making soil, digesting pollutants, nourishing and killing plants, surviving in space, inducing visions, producing food, making medicines, manipulating animal behavior, and influencing the composition of the Earth's atmosphere. Fungi provide a key to understanding the planet on which we live and the ways that we think, feel, and behave. Yet they live their lives largely hidden from view, and over 90% of their species remain undocumented. The more we learn about fungi, the less makes sense without them. Fungi make up one of life's kingdoms, as broad and busy a category as animals or plants. Microscopic yeasts are fungi, as are the sprawling networks of honey fungi or Armillaria, which are among the largest organisms in the world. The current record holder in Oregon weighs hundreds of tons, spills across 10 square kilometers, and is somewhere between 2,000 and 8,000 years old. There are probably many larger, older specimens that remain undiscovered. Many of the most dramatic events on Earth have been and continue to be a result of fungal activity. Plants only made it out of the water around 500 million years ago because of their collaboration with fungi, which served as their root system for tens of millions of years until plants could evolve their own. Today, more than 90% of plants depend on mycorrhizal fungi, from the Greek words for fungus, mykes, and the root rhiza, which can link trees in shared networks, sometimes referred to as the wood wide web. This ancient association gave rise to all recognizable life on land, the future of which depends on the continued ability of plants and fungi to form healthy relationships.
Plants may have greened the planet, but if we could cast our eyes back to the Devonian period, 400 million years ago, we'd be struck by another life form, Prototaxites. These living spires were scattered across the landscape. Many were taller than a two-story building. Nothing else got anywhere close to this size. Plants existed, but were no more than a meter tall, and no animal with a backbone had yet moved out of the water. Small insects made their homes in the giant trunks, chewing out rooms and corridors. This enigmatic group of organisms, thought to have been enormous fungi, were the largest living structures on dry land for at least 40 million years, 20 times longer than the genus Homo has existed. To this day, new ecosystems on land are founded by fungi. When volcanic islands are made or glaciers retreat to reveal bare rock, lichens, pronounced lichen, a union of fungi and algae or bacteria, are the first organisms to establish themselves and to make the soil in which plants subsequently take root. In well-developed ecosystems, soil would be rapidly sluiced off by rain, were it not for the dense mesh of fungal tissue that holds it together. There are few pockets of the globe where fungi can't be found, from deep sediments on the seafloor to the surface of deserts, to frozen valleys in Antarctica, to our guts and orifices. Tens to hundreds of species can exist in the leaves and stems of a single plant. These fungi weave themselves through the gaps between plant cells in an intimate brocade and help to defend plants against disease. No plant grown under natural conditions has been found without these fungi. They are as much a part of planthood as leaves or roots.
The ability of fungi to prosper in such a variety of habitats depends on their diverse metabolic activities. Metabolism is the art of chemical transformation. Fungi are metabolic wizards and can explore, scavenge, and salvage ingeniously, their abilities rivaled only by bacteria. Using cocktails of potent enzymes and acids, fungi can break down some of the most stubborn substances on the planet, from lignin, wood's toughest component, to rock, crude oil, polyurethane plastics, and the explosive TNT. Few environments are too extreme. A species isolated from mining waste is one of the most radiation-resistant organisms ever discovered, and it may help to clean up nuclear waste sites. The blasted nuclear reactor at Chernobyl is home to a large population of such fungi. A number of these radio-tolerant species even grow towards radioactive hot particles and appear to be able to harness radiation as a source of energy, as plants use the energy in sunlight. Of the hyphosphere. Since life first appeared on Earth, fungi have watched over the world's evolution. As novel life forms arose eons ago, the fungi found new habitats to dwell within and support. And as those beings died, the fungi around them helped recompose their bodies into the next generation. Weaving into fields and forests, the fungal queendom spread across time, holding on to ancestral forms that retain the same potency as they did long ago. Today, this reach has become so vast that fungi comprise the largest and most ancestral group of eukaryotic organisms in the mycelium of life. If the entirety of the Earth's history was compressed into a 24-hour day, and today was midnight on that clock, fungi may have evolved as long as 18 hours and 40 minutes ago. In comparison, modern humans would have only existed for 1 second. As human cultures arose and developed in this short period, the wise and elder fungi led the way. Where beans and corn were planted to feed humans, fungi hid in the soil to assist in that nourishment. And as seeds traveled the world, fungal blights followed as hidden reminders of nature's power over humanity's fate.
Pansporia prototaxites and paleomycology. Wow. Soon after Earth condensed from a cloud of space dust 4.5 billion years ago, BYA, it was struck by Theia, a protoplanet approximately the size of Mars. Ripping 20% of Earth's mass into orbit, this powerful collision gave rise to 2 small moons that eons later fused to become the current moon, a silver orb born in a dichoroidization of the night sky. Down below, Earth's oceans slowly cooled as its solid material condensed into Pangaea, a great landmass of sterile rock. Around 0.4 to 1 billion years later, cell-based life arose. It is generally assumed that the first cell was a simple prokaryote bacterium, though some researchers suggest that bacteria actually descended from the more complex cells of eukaryotes. Regardless of its initial form, the exact time, place, and means by which life developed is unclear and heavily contested. The most commonly accepted evolutionary models claim that a perfect blend of chemicals floating in an ocean, pond, or the Pangaean atmosphere accidentally collided and formed proteins capable of reproducing RNA, DNA, and a cell membrane to contain it all. Whether
of extra, oh, go ahead.
No, go ahead.
Whether of extraterrestrial or homegrown origins, ancient fungi do appear to have been present in the earliest epochs of our planet. However, putting a finger on exactly when they first develop is challenging. Compared to plant and animals, fungal tissue does not really fossilize, leaving them poorly represented in the fossil record. Very strong evidence of fungal growth in 3.5 billion years old volcanic glass from the ocean floor, and undescribed filamentous fungi have also been found in ancient oceanic basalt deposits. The vast array of chytrids, Ascomycota, and basic mycota found in deep sea thermal vents even suggest that the major fungal phyla diversified long ago in the ocean and not on land as it's commonly assumed. Indeed, the ocean may hold the keys to understanding the historical and ecological origin of fungi. But unfortunately, mycologists and marine biologists have largely overlooked marine fungi, leaving our current knowledge of their origin extremely limited. Whether born in the sea or on land, life for the first eukaryotic cells must have been incredibly challenging. On barren Pangaea, the only nutrient sources for these organisms would have been organic compounds, rock minerals, and the accumulating bodies of dead microbes. And in marine environments, the oxygen-poor and sulfide-rich ocean would have limited life to anaerobic creatures. The first eukaryotes would therefore have only been able to live if they could digest rock, eat dead organisms, and survive in anaerobic or otherwise harsh environments. When we look at modern fungi, we find all these traits, as well as many others, that would have conferred great fitness under these extreme conditions. As many researchers have posited, it can therefore only be concluded that fungi were the first eukaryotic life forms on the planet. In other words, the fungal kingdom is the grandmother of all plants and animals on Earth today.
Largely constricted to the shores of Pangaea, these early lichens set the stage for later life forms to thrive. By demineralizing rocks and digesting the tissues of their deceased brethren, these proto-lichens and ancient fungi slowly penetrated the interior of the vast lithosphere and created the first soils of Earth. Over time, the algae and proto-lichens grew larger and more complex, eventually turning into the organisms we now call plants. As plants developed, some of their fungal partners separated and spread across the terrestrial plane, while many others traveled inside the plant tissues, becoming endophytes. The simple rootless plants known as hornworts may represent an intermediate stage in this transition, as their simple algal-like bodies host endophytes that curiously form arbuscule-like structures. Endophytic fungi seem to have been found in hornwort fossils dating from the Phanerozoic era, about 540 MYA. As these and other early plants evolved, like the ancient genera Rhinospethia and Horneophyton, they took on simple bifurcating structures that resembled the growth patterns of the endophytes inside them. Many of these early plants spread by means of spores.
This was an explosive era for evolution. As fungi and bacteria continued to build Earth's soils, a massive lichen-like organism began to appear on shorelines, towering over the insects and plants below. At up to 3 feet wide and 26 feet tall, these species from the genus Prototaxtes were by far the largest organisms on the planet when they first appeared around 420 million years ago. And considering their significant abundance and diversity, they seem to have also been some of the most successful species in their habitat. Prototaxtes existed for about 70 million years until around 350 million years ago, more than 3 times as long as the genus Homo has existed. With an anatomy unlike any living or fossil organism, Prototaxtes specimens have been historically hard to classify, and various attempts to place them into evolutionary models have been repeatedly challenged. In cross-section, their fossils have concentrically arranged sections that are so similar to tree rings that specimens were originally thought to be small coniferous trees. However, in 2007, tests proved that due to the wide range of carbon types in their fossils, Prototaxites must have been a perennial fungus-based decomposer and a major recycler of organic life in their environment. Further, as the majority of Prototaxites specimens are made of 3 types of interwoven hyphae that host clamp connections, their tissue may have been very similar to trimitic woody polypores from the Basidiomycota. Cavities in them seem to have been bored by insects, suggesting the giant organism was a refugee for bugs, while their exterior was likely coated in algae, making more like a giant lichen than a mushroom.
Along with its classification, the shape of Prototaxites is also contested. For though this ancient sentinel is often depicted as a smooth column in contemporary artistic representations, it may have produced branches with large scale-like leaves, giving it an appearance similar to a conifer tree. As plants and animals continued to evolve, so too did their fungal ancestors. At some point, species in the Ascomycetes and Basidiomycetes began forming fleshy mushrooms, though we did not know if gilled or poroid fruit bodies developed first. Currently, the oldest mushroom fossil is an extinct marasmius-like species, Archomarasmius legetleti, that has been found in 90 to 94 million year ago amber. After the Permian Triassic extinction event, 251.4 million years ago, the fossil record shows an increase in fungal spore counts, suggesting that fungi proliferated on the abundance of dead plant and animal matter. Around the time of the dinosaurs, the Mesozoic era, circa 252 million years ago, plant-fungal symbioses took another leap, forming the ectomycorrhizal relationship. However, the best evidence of ancient ectomycorrhizal fungi comes from Rhizopogon and Sulius-like specimens dating to around 50 million years ago. It wasn't until around 100 million years ago that ericoid mycorrhizae evolved. Similarly, as animals adapted to their changing habitats, they maintained an increasingly intimate link with fungi. Yeast-associated insects survived through extinction events, while later herbivores thrived by carrying fermenting fungi in their guts, just as cows host chytrids today. The proliferation of fungal spores from the post-KT extinction fossil beds, circa 66 million years ago, point to another period of post-apocalyptic fungal dominance. This is yet another reminder that even in the darkest hour, following the loss of most of the world's inhabitants, fungi will remain to recompose the fallen and carry life into future eras.
In the Carboniferous period, 290 to 360 million years ago, the earliest wood-producing plants spread across the tropics in swampy forests, supported by their mycorrhizal fungal partners. These forests grew and died, pulling huge quantities of carbon dioxide out of the atmosphere. And for tens of millions of years, much of this plant matter did not decompose. Layers of dead and unrotted forest built up, storing so much carbon that atmospheric carbon dioxide levels crashed, and the planet entered a period of global cooling. Plants had caused the climate crisis, and plants were hit the hardest by it. Huge areas of tropical forest were wiped out in an extinction event known as the Carboniferous rainforest collapse. How had wood become a climate changing inducing pollutant? From a plant perspective, wood was and remains a brilliant structural innovation. As plant life boomed, the jostle for light intensified, and plants grew taller to reach it. The taller they became, the greater their need for structural support. Wood was plants' answer to this problem. Today, the wood of some 3 trillion trees, more than 15 billion of which are cut down every year, accounts for about 60% of the total mass of every living organism on Earth, some 300 gigatons of carbon. Wood is a hybrid material. Cellulose, a feature of all plant cells, whether woody or not, is one of the ingredients and the most abundant polymer on Earth. Lignin is another ingredient and the second most abundant. Lignin is what makes wood wood. It is stronger than cellulose and more complex. Whereas cellulose is made up of orderly chains of glucose molecules, lignin is a haphazard matrix of molecular rings. To this day, only a small number of organisms have worked out how to decompose lignin. By far the most prolific group are the white rot fungi, so-called because in decomposition, they bleach wood a pale color. Most enzymes, biological catalysts that living organisms use to conduct chemical reactions, lock onto specific molecular shapes. Faced with lignin, this approach is hopeless. Its chemical structure is too irregular. White rot fungi work around the problem using non-specific enzymes that don't depend on shape. These peroxidases release a torrent of highly reactive molecules known as free radicals, which crack open lignin's tightly bonded structure in a process known as enzymatic combustion.
Fungi are prodigious decomposers, but of their many biochemical achievements, one of the most impressive is this ability of white rot fungi to break down the lignin in wood. Based on their abilities to release free radicals, the peroxidases produced by white rot fungi perform what is technically known as radical chemistry. Radical has it right. These enzymes have forever changed the way that carbon journeys through its earthly cycles. Today, fungal decomposition, much of it of woody plant matter, is one of the largest sources of carbon emissions, emitting about 85 gigatons of carbon to the atmosphere every year. In 2018, the combustion of fossil fuels by humans emitted around 10 gigatons. How did tens of millions of years worth of forests go unrotted over the Carboniferous period? Opinions differ. Some point to climate factors. Tropical forests were stagnant, waterlogged places. When trees died, they were submerged in anoxic swamps where white rot fungi were unable to follow. Others suggest that when lignin first evolved in the early Carboniferous period, white rot fungi weren't yet able to decompose it and required several million more years to upgrade their apparatus of decay. So what happened to the vast areas of forests that didn't decompose? It's an inconceivably large amount of matter to pile up, kilometers deep. The answer is coal. Human industrialization has been powered on these seams of unrotted plant matter, somehow kept out of fungal reach. If given the chance, many types of fungi readily decompose coal, and a species known as the kerosene fungus thrives in the fuel tanks of aircraft. Coal provides a negative of fungal histories. It's not a record of fungal absence of what, oh, excuse me, it's a record of fungal absence of what fungi did not digest. Rarely since then has so much organic material escaped fungal attention.
The mycorrhizosphere. Our exploration of fungal systems begins with a cross-section of soil, the hidden ecology. Underground and subcultural, soil communities build the world we stand upon and sustain the life that fills all biomes. They are the earthly womb from which life springs and the dark tomb to which all things return. A universe of unfathomable complexity, the soil is filled with innumerable organisms performing invisible acts that, despite countless influences, somehow remain finely balanced and forever abundant. Soils are the history books of the world encoded by fungi, microbes, and insects into each chapter of leaf and rock. To read this story, the soil requires us to examine the unknown, to seek subtleties in expression, and to uncover complexity through a blend of sharp awareness and the ability to wonder. Without offering the soil the chance to unveil its countless dynamics, it will only appear as a dead, static mass of minerals, the way it has to scientists for the bulk of agriculture's modern history. With over twice the biomass of all of Earth's animals, soil is truly alive and can, in a sense, be regarded as a sort of massive organism. Like all living beings, the soil takes in, transforms, moves, and releases nutrients for other organisms to consume. But, more critically, the soil is the central axis in these nutrient cycles. It is through soil that carbon, nitrogen, and many other elements complete their resurrection from decay back into living matter. The soil is the bridge that links the largest organisms with the smallest microbes, just as the air we exhale is endlessly inspired by other animals and plants.
Soil cannot be created in a lab, nor should it be seen as the sum of its components. Indeed, the vast majority of soil organisms cannot be intentionally cultivated, their lives being inextricable from soil body. The habitat of these microbes, the inanimate aspect of soil web, is ancient, being born over eons by the slow reduction of organic matter to small substances, collectively known as humus. Humus takes such a long time to accumulate that it may only reach a depth of 1 meter in an old growth forest, while it is often only an inch or 2 deep in developed areas. With such a small amount of topsoil on the planet, this protective layer of the earth must be spoken for, understood, and held sacred by all those who care for the wild. It is the skin of the world, tattooed with the legacies of its inhabitants. But just as our skin can be easily wounded, so too is the soil readily broken and scarred if not defended from injustice. To talk of any ecology or its protection is to speak of soil. So precious is the connection to the web of life. As with most soil microbes, the diversity, distribution, and dynamics of soil fungi are barely understood. Soil yeasts offer a significant contribution to nutrient cycles and also provide a food source for other microbes and insects. Yet, to date, they have received limited attention by ecologists or mycologists. The microparasitic Trichoderma species are highly abundant in soils performing a critical check and balance function amongst soil fungal populations. And yet mushroom growers who don't realize their importance to come to despise these species for their unwanted growth on mushroom mycelium in the lab. The soil fungi that are by far the best understood are those that form a mutually beneficial relationship with living plant roots. Through the entanglement of their hyphae with delicate root hairs, various fungi from multiple phyla form a mycorrhizal symbiosis, a fungus plant dynamic that is shared by more than 6,000 fungal species and at least 90% of the world's plants. Mycorrhizal fungi are found in all habitats and latitudes, permeating the soil matrix. Where wild soils remain intact, a single gram of earth may contain 1,000 to 10,000 fungal propagules and a mile or more of hyphae. A single acre in an aged Douglas fir stand can contain 3,700 pounds of mycelium by dry weight and 4,800 pounds of mycorrhizal rootlets. This ubiquity results in a wide range of benefits to not only the organisms involved, but to the entire ecosystem in which they are enmeshed.
The
The
point of
point-
connection. Go ahead.
It's either way. All right. The point of connection between two organisms, the mycorrhiza, is not defined by the fungus or the plant that the fungus inhabits. Rather, mycorrhiza is the singular symbiotic partnership of the two beings. Mycorrhizal structures come in several forms, though all hold the root hypha intersection as their central feature. In all instances, the mycelium wraps around the plant's root tips and enters the root cellular structure to a depth of at least the first few cell layers. Typically, mycorrhizae develop on undifferentiated root hairs, meristems, where the plant absorbs the bulk of its water and nutrients. Some mycorrhizal fungi only enter the root to a shallow depth. They are the ectomycorrhizal species, many of which form edible and medicinal mushrooms or truffles. Other species travel deeper into the root structure and penetrate the walls of root cells. These are known as endomycorrhizal fungi. Some endomycorrhizal fungi do not form mushrooms, but live cryptic lives beneath the soil horizon. In all mycorrhizal structures, nutrients are passed between the two partners. The mycorrhiza is like a barter station, where each species provides something of value that the other could not obtain on its own. With its ability to photosynthesize, the plant reaches towards the sun and creates sugars that it sends down to the fungus. In return, the slim and quick-growing hypha extend far into the soil matrix to draw in water, nitrogen, phosphorus, and other nutrients that the thick and slow-growing root hairs would not be able to easily reach on their own. In effect, the fungus expands the absorptive area of the plant roots 10 to 1,000 fold, thereby increasing the growth rate and survival times of the roots, while decreasing the time it takes for the plant to reach maturity. The benefits of this enhanced nutrient access include a higher nutritional content, medicinal quality, and flowering rate in mycorrhizal plants, which compared to non-mycorrhizal plants of the same nursery stock.
This increase in absorptive surface area is not the only way that these fungi enhance nutrient access. If it were, plants would likely have evolved to produce longer or slimmer roots long ago, fungi produce a unique array of digestive enzymes and acids that are able to unlock nutrients that would otherwise be unavailable to plants. These nutrients may come from organic carbon-containing substances, such as plant and animal debris, or they may be bound in inorganic, non-carbon-containing substances, such as rocks and clay. Through their digestive abilities, fungi transform these nutrients from a non-soluble or unavailable form into a water-soluble or available form that plants can readily take up into their tissues and metabolize. The fungi transmute inert substances into nourishment, channel these compounds through their mycelial networks across the soil, and provide them to whole plant communities and ultimately to the entire ecosystems that the plants inhabit. And when these plants die, they are churned back into the soil matrix by decomposing fungi, where their nutrients eventually pass on to other organisms. Unable to access distant or non-soluble nutrients, many plants, for example, grapes, citrus, melons, oaks, and pines, fail to survive without a mycorrhizal partner. This is because most plant roots have not evolved to perform the chemical transformations that release soil-bound nutrients. Roots are essentially porous anchors that primarily stabilize the plant and serve as a beacon and platform for the beneficial microbes and fungi that actually produce and provide much of the plant's nutrition. Where mycorrhizal fungi are absent, most plants must be fed with water-soluble fertilizers to obtain their basic nutrition. However, the extraction, industrial processing, and transglobal shipment of both artificial and natural fertilizers incurs a range of significant and hidden social and ecological costs, making them unsustainable options for enhancing crops. Fertilizers also tend to be applied in excess, resulting in a significant loss of nutrients that wash past root zones. These excess fertilizers then leach into ground and surface water systems, where they disrupt soil ecologies and flood into river deltas to feed blooms of toxic algae that choke out other aquatic life. Problems such as these can be avoided in food production systems through the use of compost and the cultivation, inoculation, and stimulation of mycorrhizal fungi.
This relationship has numerous other effects. As mycorrhizal fungi are able to tolerate lower water levels than most plants, these species can reduce a plant's dependency on constant water access, thereby reducing the drought stress that would otherwise cause non-mycorrhizal plants to wilt and die. This is especially important in desert ecosystems where cacti and most other plants rely heavily on mycorrhizal fungi for their hydration and survival. Mycorrhizal fungi also provide plants with protection from soil pathogens. The mycelial coat around the root acts as a protective shield against antagonists, while the fungus may also release antibiotic compounds into the mycorrhizosphere that are targeted to specific pathogens. Mycorrhizal fungi can also indirectly suppress pathogens by competing for nutrients with the production of chelating siderophores. By influencing the plant's production of its own endogenous defense mechanisms, increasing a plant's production of lignin, for example, or by stimulating the growth of other beneficial microbes. In effect, mycorrhizal fungi not only help increase the plant's overall resilience against attack, They also reduce the need for plant growers to apply natural or artificial pesticides that disrupt the delicate dynamics of soils. Through mycorrhizal relationships, fungi unite the ecologies of the world. Where soils remain undisturbed, these partnerships do not remain limited to the combinations of one or multiple fungal species and a singular plant. Mycelial networks can connect the roots of dozens of plant species across phyla within an ecosystem. They are the connective tissue that ties whole ecosystems together through a common mycelial network, in which nutrients, water, and information are constantly exchanged between organisms large and small. In dense forests, mycorrhizal fungi have been shown to carry sugars produced by elder trees to seedlings that would otherwise die in the dense shade of the understory. In one study, a similar type of nutrient sharing was shown to occur between a Douglas fir tree and a paper birch, first in one direction and then in the other.
One of the ways fungi might help save the world is by helping restore contaminated ecosystems. In mycoremediation, as the field is known, fungi become collaborators in environmental cleanup operations. We have recruited fungi to break things down for millennia. The diverse microbial populations in our guts remind us that in those moments in our evolutionary history, when we haven't been able to digest something by ourselves, we've pulled microbes on board. Where this has proved impossible, we've outsourced the process using barrels, jars, compost, heaps, and industrial fermenters. Human life hinges on the many forms of external digestion using fungi, from alcohol to soy sauce, to vaccines, to penicillin, to the citric acid used in all fizzy drinks. This sort of partnering, in which different organisms together sing a metabolic song, neither could sing alone, enacts one of the oldest evolutionary maxims. Mycoremediation is just a special case.
It shows great promise. Fungi have a remarkable appetite for a range of pollutants besides toxic cigarette butts and the herbicide glyphosate. In this book, Mycelium Running, Stamets writes about a collaboration with a research institute in Washington State, which partnered with the US Department of Defense to develop ways to break down a potent neurotoxin. The chemical dimethylphosphonate, or DMMP, was one of the deadly components of VX gas manufactured and deployed in the late 1980s by Saddam Hussein during the Iran-Iraq War. Stamets sent his colleagues 28 different fungal species which were exposed to the compound in gradually increasing concentrations. After 6 months, 2 of the species had learned to consume DMMP as their primary nutrient source. One of the tramites, or turkey tail, and the other was psilocybe azurescens, the most potent psilocybin producing species known, discovered by Stamets several years before and named for the bluish hue on the stems. He later named his son Azurus after the mushroom. Both are white rot fungi. The mycological literature is filled with hundreds of such examples. Fungi can transform many common pollutants in soil and waterways that endanger lives, whether human or otherwise. They are able to degrade pesticides such as chlorophenols, synthetic dyes, the explosives DNT and RDX, crude oil, some plastics, and a range of human and veterinary drugs not removed by wastewater treatment plants, from antibiotics to synthetic hormones.
In principle, fungi are some of the best qualified organisms for environmental remediation. Mycelium has been fine-tuned over 1 billion years of evolution for one primary purpose: to consume. It is appetite in bodily form. For hundreds of millions of years before the plant boom in the Carboniferous, fungi made a living finding ways to decompose the debris that other organisms left behind. They can even boost decomposition by providing mycelial highways that allow bacteria to travel into otherwise inaccessible sites of decay. And yet, decomposition is only part of the story, excuse me. Heavy metals accumulate within fungal tissues, which can then be removed and disposed of safely. The dense meshwork of mycelium can even be used to filter polluted water. Mycofiltration removes infectious diseases such as E. coli and can sop up heavy metals like a sponge. A company in Finland uses this approach to reclaim gold from electronic waste. Is that it?
That's it. Thank you, Ansu, and thank you everyone who listened and also read. Yes, that was a very long reading and a little dense, but we have James here. If anybody has any questions, you guys can ask him. The mycorrhizal network are countless assemblages or thin threads that lie underneath with complex soil communities from which springs forth the life we see above our feet. We too become part of that network. In some ways, the mycelium network is the best example of the more than human infrastructure. And now we would like for Mish to share our guiding questions on the chat bar, and also if you can speak them out loud, Mish. There you are. Thank you.
Yep. All right. So I know the questions were posted a little bit early on, but I'm still going to put them again. So first, there is a short prompt, which reads as such: FarmLab investigates and works with a queendom of fungi on our site through microscopy, inoculating logs, and culturing fungi. First set of questions: how do we interact with invisible webs and gardens, such as mycorrhizal network? How do we observe, hold space for, appreciate, and recognize living systems that are always felt but not directly seen? And the second set of questions is: how do you engage with decomposition metaphorically and literally? Do you have a cycle of breaking down your practices, emotions, beliefs, experiences, and relationships? What is the fruiting body or bounty that follows in its wake? And I will put this now in the chat bar. Please feel free to respond directly in the chat bar if you don't want to speak during the group. And there it is.
And now I will move everybody into breakout rooms for about 12-15 minutes, and we will see you back here soon.
Well, I've
Welcome back.
Welcome, sorry, go ahead, Jen.
I was just saying, welcome back.
Welcome back, everyone. And I know we understand it is after 7 and we're running into that time. So maybe we can briefly just go around and summarize what our conversations were, and then we can feature the Beaver of the Week with Emily. Thank you.
Millie, do you want us to do a quick wrap up of the groups or?
Yeah, sure, go for it.
Okay. No, I just interestingly in the group, we all had one thing in common. It was actually about mycelium and fungi and music for somehow. And so Lily put some links in the chat in the chat bar, and also Alexandra and even Carolina. So it's interesting that we all converge all of a sudden. And I was telling how 20 years ago we could have had the similar conversation about the compost, but not about fungi and mycorrhizas. This is just something that now is emerging. We're barely touching on something that has evolved for the last, I don't know, many billion years, and that is daunting to me. That's all. And we didn't have enough time anyway.
Lee just left, but she was imparting a lot of wisdom around the use of mycelium for growing vegetables and maintaining trees during droughts. And yeah, unfortunately cut us off just as she was giving some valuable information about leaving the white rot fungus that grows on the squash that we have in our yard. But when I was doing some digging to put our vegetable garden in, I was wondering why all the fruit trees that have been there for decades have been okay with the really bad irrigation system that our building has. And because the water runs away from the trees and doesn't sink into soil. So it seems like it's also those mycelial networks that came up when I was digging are sustaining those trees for the long periods of time when the water doesn't actually sink into their roots.
Did she also give you the tip of aerating your soil and maybe also adding compost?
She did, and also to not till. She reminded us about how plowing is like this 12,000-year-old error that we just keep ruining the skin of our earth by tilling it every harvest.
Absolutely. Thanks for sharing, Adam.
Well, our group really focused on the unseen world, and we talked about how being exposed to twinkling stars under a no moon night is shocking and surprising because they're always there every day, but somehow it's only when you see them that you feel like they're present. So if that's true, then why wouldn't the undernet of all of the living things not be as complicated and abundant as that the heavens are? And we talked about the miracle of attending the birth of a baby, when 1 minute there's a new, in a second there's a whole new human being in a room. And they were there before, but they weren't there before, and then all of a sudden they're there before and they're perfect and whole and completely themselves. And and so ultimately, Ansu was articulating in her inimitable way that perhaps this is an epistemological switch, right? That's being made, that it's the unseen that is the thing to be aware of. I butchered your statement, but
oh no, I think it was that science, the bias of science is always to have things that are tangible and measurable and quantifiable, and this whole world of the fungi leads us to use other ways of knowing, and if it gets at a much deeper truth about who we are, this continuous ancient lineage.
Yeah, you said an epistemological shift embodied, which I thought was so beautiful.
You know, that's one of my favorite things about mycorrhizae is that it's, you know, kind of defying the taxonomy slicing and dicing of our world into, like, you know, all these terms, phyla, species, genus, things like that. And, you know, mycorrhizae are neither the plant nor the fungus. And what we're seeing in this case is it's also even a step further. It's neither the singular plant or the singular fungus if it's connected in this entire network. And this is one of the most important and significant relationships in the history of life on Earth, and that it is completely antithetical to taxonomy in many ways. Because yeah, like you said, it just immediately goes into that other web of knowing.
Well, thank you, everyone. Thank you, everyone who read, who participated tonight. Just wanted to remind everyone that next week we're going to take a break for those of us to go out and vote here in Los Angeles. And we will return on November 15th. And we will have the entire Farm Lab team on board next week. And we will be reading an excerpt from our colleague Diego Zapata on seeds, and we'll be available to answer any of your questions or even talk about the series of post-human infrastructure. So thank you everyone again, and have a great evening. And for those of you that will be celebrating the Dia de los Muertos, have a great evening with your ancestors.
Are we going to show a film, Millie?
My bad. So sorry. Emily, show us the beaver of the week. Thanks for the reminder, Lauren.
Yay. Yay.
For some reason, I was looking at prehistoric beavers and I just put the image in the chat bar. It's pretty cool.
All right, friends, I am going to now do the ofrenda and I will see you guys tomorrow.
Good night.
Bye-bye. Good night. Ciao,
Good
ciao.
night. Ciao, ciao.
Bye.